Hello, College Biology. This is our first pre-lab video for our enzyme activity lab, and we're going to start this discussion at the organelle level right here. There are lots of organelles, lots of different cell types. Today we're going to focus on the peroxisome, and what the peroxisome does is it produces hydrogen peroxide from the detoxification of various substances. That's kind of like a backwards description of what it does. It helps break down fats. It helps break down toxins. These are good. Fats can be a source of energy, so breaking those down so they can be recycled and get energy out of them, that's great. And then toxins that would kill the cell, like alcohol or other problems, want to get rid of them so they don't cause problems. That's great. But they produce hydrogen peroxide as a waste product of that detoxification. Well, the hydrogen peroxide itself causes problems. We use hydrogen peroxide as a cleaning agent because hydrogen peroxide kills cells. So in removing the toxins, we create essentially another toxin. We create something that is also bad for the cell. So we got to get rid of that. And so to do that, the peroxisome also creates an enzyme called catalase. Catalase breaks down hydrogen peroxide in the formula you see here. It'll take two hydrogen peroxide molecules, rip them apart, rearrange the atoms to create two water molecules and one oxygen molecule. Every organism with a nucleus makes catalase, including the easily obtainable yeast. Yes, this is active dry yeast, the kind used in making bread. So yeast makes a lot of catalase and it secretes it into its surrounding environment. So that way it just sort of, instead of waiting for something to invade the yeast cell or toxin to get into the yeast cell, it puts the catalase out in the environment so that if there are toxins approaching the yeast cell, they get digested ahead of time. So great. Thanks, yeast, for making being our source of catalase. Well, what does this have to do with our lab? Well, if you take a filter paper disc and coat it in catalase made by yeast and drop it in hydrogen peroxide, it will do that reaction. It will take the hydrogen peroxide, all of this liquid that's surrounding the disc, and it will rip it apart and create water and oxygen gas. So if you let it sit in there for an amount of time, oxygen gas bubbles will build up on the disc, and then if with enough time, those oxygen gas bubbles will reduce or increase the buoyancy of the disc, and it will float to the top. Well, if catalase isn't working and you put the disc in hydrogen peroxide, well, then it's not going to do that reaction. So it doesn't rip apart the hydrogen peroxide. It doesn't make water. It doesn't make oxygen gas. So it doesn't matter how long you wait. If the catalase does not work, you will not digest hydrogen peroxide. You will not get oxygen gas, and it won't float. And there is also a middle ground. If some of the catalase is working, not as much as when everything's in good condition, but if some are working, then some of them will make bubbles, but it'll take a lot longer to make enough bubbles to get the disc to rise. So how long it takes the disc to rise is a proxy for how well the catalase is working. And you're going to use that information in your lab. So I hope this helps. Struggle well.